Welcome everyone, and thank you so much for joining us for today's bio talk on biomanufacturing, health and medicine, or District 2 Bio Talks. For those of you who's the first time that are attending, what we do is we provide a spotlight on what's going on the life sciences sector from a point of view of experts, like we're doing today. In most of these cases, these experts will be scientists that are also entrepreneurs, and we want to really showcase their work. So for today, we have two veterans in scientific entrepreneurship. Each of them has over 30 years of experience working on research spin-offs. And today, we have the pleasure to have them with us. They will be sharing their experiences as entrepreneurs, as scientists, but they will also share this perspective on the future of biomanufacturing, as well as the very exciting work they're both doing on COVID-19, either on therapies or vaccines. So while we wait for a couple of other participants to arrive, we started quite early today, thanks to Rosa. So I'll just wanted to run a quick survey. So Rosa, if you could help me with that. You will be seeing a survey appearing on your screen, and will be appreciated if you could just fill that in. It's just to learn a little bit more about you. Right, and while that is being completed, just let me introduce myself. My name is Anna, and I'm a coordinator of life science initiatives at District 3. Myself, I'm a scientist as well, and I guess in the pool that you're currently seeing, I'm in the category of the wanna be an entrepreneur, so I'm always very open to ideas and new projects. And I'm gonna be your MC for today. So what will happen today is we're gonna go through about 30 minutes of questions guided by me, and then we'll open the panel to answer questions from the audience. So please join the conversation by adding your questions and comments through the chat. You can already start writing those questions if you have some of them already. Otherwise, feel free to write them later as our panelists speak. So I'll introduce briefly District 3 as well before we start. So for those of you who are not familiar with us, we are a leading innovation hub based at Concordia University, and we support technology-based teams to create businesses with global impact. Our services are open to everyone. You don't need to be associated with Concordia University to join District 3, and our services are also for free. So it's a really great opportunity for those of you who are scientists or entrepreneurs and want to join. I send my email, I show my email before, so feel free to reach out if you have any questions about that. Since we've been around, we've worked with about 530 startups, and approximately 30% of those are startups in the life sciences and biomanufacturing. So this is really just a snapshot of our community of District 3 in life sciences and biomanufacturing. As I mentioned, it's about 30% of all the startups we work with. And I guess for today, I just want to highlight IntelliStem Technologies, who's also working on a COVID-19 vaccine development with very promising results in a preclinical stage. We have other different startups that work on emerging technologies, most of them, for example, microfluidics or synthetic biology, as it's the case for Dropgenie or Hyacinth Bio. And something that is very exciting is the fact that we are going to be able to keep on growing our life sciences community even more. And this is thanks to the 52 million investment that has been done by Concordia University, as well as provincial and federal governments, that has allowed us to open the BioHope. The BioHope will be the Montreal first incubator and accelerator for life sciences, and will have a laboratory and co-working space as well. Sorry. So, and if this is what I'm saying, it sounds really interesting to you and you wish that you wanna join our community of life scientists working in biotechnology, but you don't have a specific idea or project yet that you wanna work on, there's something I wanted to talk about right before we start, which is the Quebec Scientific Entrepreneurship Program. It was a program developed with District 3 and the Fonds de Recherche de Québec, and it's designed to help scientists have a taste of what it's to be an entrepreneur and working in innovation. So this program is completely free, online, and bilingual. And it will take you over 3 different phases where you're going to be able to launch your startup at the end of it. You start by a discovery phase and ideation phase where you can see how you can bring your research to the market. So I would highly encourage you to look at that if that's something that interests you. And the nice thing about this program as well is that it allows you to join a huge community of scientists working in the life sciences. Okay, next slide. So let's go back to today's topic. And really, at District 3, having this large community of life scientists, we know that everyone is wondering how the COVID-19 pandemic can potentially really lead to lasting transformation in investing and collaborating, also in regulations, in the biotechnology sectors, and all the different collaborations that we can have between universities, scientists, and the industry. And this is exactly why we have put this panel together today. So I'll have the pleasure of welcoming Dr. Ward and Dr. Nigh. Let me start by introducing Dr. Ward. He wears multiple hats. Besides being a professor of experimental medicine at McGill University, he's also the medical officer of Medicago. So you probably heard that Medicago received a $7 million support from the government of Quebec for its work on the COVID-19 vaccine. And they have also very recently announced positive results in their animal tests. What I like to highlight about Medicago is that they started back in 1997 thanks to a partnership between Laval University and Agriculture Canada. Again, highlighting the importance of collaboration collaboration and crosstalk between universities, governments, and industry, and really the necessity for long-term investment in science. So Dr. Ward, he also likes to dispel myths and taboos between what it is to work in academia and industry. And he's also working on a new research spin-off that he will be telling us more about today. So thank you so much, Dr. Ward, for joining us. And I guess I'll start by asking you about your career path. So maybe you can give us a brief overview about your career path and specifically what made you take the decision of embarking in endeavors outside academia.
Well, so thank you very much for inviting me. It's a topic of some interest to me. My career path was interrupted several times by me hating what I was doing and quitting. And so I left medical school after the 3rd year and then came back under protest, left my fellowship and did other things that interested me more, and eventually came back. Completed my medical degree and training, but one of my mentors in that last stage, Dr. John Bartlett at Johns Hopkins, made the point that if I was going to embark on a research career in academics, I was going to go through periods of time where the funding was not as generous as it was at other times, and that I should always try to have at least one industry contract or collaboration that I was working on, because it would keep my research lab alive during the dry times, and in the flush times, it would be something extra that I could do. That was awfully good advice, and so from a very early stage in my career, I made an effort to accept opportunities that came my way with different industries. Whether that was in diagnostics or drug development, or most particularly in vaccine development, and that's been the focus of my research activities. So I was open from a very early stage to the idea that I would work with industry, and while some of those industry collaborations have worked out brilliantly, others have been frankly disastrous. And you know, when something like that happens and it goes away, you can be very frustrated by the fact that you've spent 10 years working on something. But companies come and they go, and you have to be prepared. You can't put all of your eggs in one basket. So my career has been largely spent in academics, where I've had a fairly large lab that's been grant driven for over 30 years now. I run the McGill Vaccine Study Center, which I've done since the early 1990s, and have established very close relationships with small and large vaccine manufacturers over the years. Mm-hmm. So I've been able to participate in vaccine evaluation at a large scale with the beer companies and vaccine development with smaller biotech startups.
That's a really cool, interesting path. I wonder, because you were mentioning many things haven't go the right way. So maybe you can tell us a little bit about what you've learned by working in these two sectors, and if you feel comfortable talking about maybe also what kind of things have gone wrong.
Oh, well, sure. I think that you have to walk into any academic industrial collaboration knowing the language. And as an academic, I walked into my first industrial collaborations where I had no idea what they were talking about. It's a completely different vocabulary. So when I was offered a position on a scientific advisory board, I was I was told that, you know, they didn't have much money, and so on. As an advisor, they could pay me a little tiny bit, but they would give me stock options. I had no idea what they were talking about. And so, you know, one of the most important things is that there really isn't a stupid question. And so I made a point of finding out what acronyms they were using and what these terms meant. Mm-hmm. That's actually served me very well, because I think most academics, Canadian American, really have no idea what they can bring to industrial collaborations. And I think there's a high level of fear among most academics in their interactions with industry, mm-hmm. because their starting position is usually that industry is going to steal their ideas and leave them with nothing, which can certainly happen. But if you know the vocabulary and you know how to interact with industry, you can certainly prevent that from happening. It's a risk that is mitigatable, if that's a word.
Yeah. Just a quick follow-up before we move on to Dr. Nigh, and it is, you said that there's kind of a little bit of friction between scientists to collaborate with the outside world. Do you see that is changing during the last years? Do you see like the students that you are dealing with now, are they more open to that, or are the universities more open to encourage this type of cross-pollination, so to say?
Well, I would like to believe that's true. I'm not sure that it really is. I do find myself in a very unusual position where I've maintained my academic role. I resigned my clinical role within the McGill University Hospital system 2.5 years ago. And the university has allowed me to retain my professorship, my lab, my office, my right to have students, and so on. So I do truly have one foot in academics and the other foot in industry, but everybody is a little bit uncomfortable with that. Mm-hmm. And you have to be, you really have to be enormously careful to be transparent in all of your dealings. Mm-hmm. And so I have a full academic profile. I'm the chair of the CIHR Institutional Advisory Board for Infection and Immunity. That's classically been held by an academic. Mm-hmm. But because I tell everybody what I'm doing, everybody knows exactly where I'm coming from. And I'm very careful to wear, you know, to say I'm putting on this hat when I'm making this comment, and I change hat and I put on the other one to make another comment. As long as you are completely transparent in what you're doing, I think that people can be comfortable, but it's not a completely comfortable position to be in still in 2020.
Of course, I imagine, and I would think that it might be even more difficult now for younger scientists that are starting their careers to do that now, given that the professorship spaces are like very small, and for them it's more difficult to be able to join that ladder in the academia. I would give those young
faculty or faculty aspirants exactly the same advice that I got from Dr. Bartlett. Pursue your academic career, but make sure that you always have, that you're always open to collaborations with industry, large and small, collaborations with smaller industries, and contracts with larger industries. If you look at the amount of money that's spent by industry on research, it dwarfs the money that is spent in the public sector. One of the myths is that industry scientists are not as good as academic scientists. That's complete nonsense. Some of the most talented scientists I've ever worked with, and I have the pleasure of calling my friends, are in industry. They are every bit as smart and they work every bit as hard as academic scientists do. So there is no discount on industry science.
Thanks. Yeah, I'm going to take that advice and say, I guess, to all the young scientists that it's never too early to start learning about things like innovation, entrepreneurship, and things that are happening outside your labs. So I'll move now to talk a little bit about Dr. Nigh, who he really did leave academia when he started his science spin-off. So Dr. Harold Nigh is joining us. I thought it was London, Ontario, but it's actually Niagara, Ontario. Is that correct? Yes. And he's the director of BLES Biochemicals, which is a biotechnology company that he started from scratch as a spin-off of his research at the University of Western Ontario. And BLES commercializes surfactant. He's going to tell us a little bit more about what that is later, and that is used for the treatment of respiratory distress syndrome in infants. And what they have been doing lately is also testing the surfactant as a potential therapy for COVID-19. They're currently running a clinical trial on that, and they're trying to see if surfactant can help improve lung function and reduce the need for medical ventilation. So among many other things that Dr. Nigh will be telling us about, he and his team have been able to grow BLES without external investors. And so thank you so much for joining us, Dr. Nigh. I will start the same way we start with Dr. Ward. So perhaps you tell us a little bit about your career path again, and also what makes you take that decision of embarking in entrepreneurship and taking this research that you were working on out of the university labs.
Well, thank you very much, Anna, for inviting me to be part of this panel discussion. I guess I'll just correct you on one or two things. I did not start the company by myself. I have a business partner. His name is Dr. David Bjarnason, and we both worked with Dr. Fred Postmeyer, who is a lipid biochemist at the University of Western Ontario. So we've been part of the team, and we are now directors, but it's not just our show. It certainly is not just my show. However, it's been a very interesting road, and yes, during my postdoc, I decided to leave academia and look for something else, and this is what turned up. So I'll talk briefly about my career path. I've made a few notes, so I'll read off a few of these things. After obtaining a PhD in chemistry from the University of Western Ontario with a specialization in physical chemistry, I spent a year engaged in postdoctoral research at the Solar Energy Research Institute in Golden, Colorado. And that's just outside of Denver, Colorado. The Solar Energy Research Institute was created by Jimmy Carter, I don't know, probably in the 80s. And I graduated in the early 90s and spent a year at the Solar Energy Research Institute. My PhD work centered around studies of biomimetic solar cells, fabricated using monolayers of an amphiphilic porphyrin molecule. In order to fabricate these monolayer structures, I needed to develop deposition systems and to build deposition systems, none of which were commercially available at the time. Consequently, I was forced to spend many hours in the grad student shop building equipment. I bring up this point because it influenced my career and it influenced the way I do my work. After returning from Colorado, I was contacted by Dr. Fred Postmeyer, who was a lipid biochemist at Western, and who was aware of the work that I had done in this area. He asked for me to build equipment for his work, which was the study of alveolar dynamics in the human lung. His research group was involved already in phase 1 and phase 2 clinical trials together with Women's College Hospital in Toronto, looking at the potential role of exogenous surfactant therapy for the treatment of neonatal respiratory distress syndrome. NRDS, as it's called, is a condition which afflicts prematurely born infants of approximately 30 weeks gestation or less who are not yet secreting their own surfactant. The role of surfactant, pulmonary surfactant, which is a mixture of lipids and proteins, which is naturally produced in the body, and we all have it in our lungs, is to reduce the surface tension of the lung alveolar surface by forming a monolayer, which through some very elegant self-assembly buttresses the alveolus against collapse on exhalation. So typically when we breathe out, each alveoli in our lung, which is a very small bubble, is at its smallest radius. And it's at that level, it's at that point where it is most subject to the potential for collapse. And what surfactant does is it prevents that collapse and allows the bubble to remain stable, just like if you blow a soap bubble as opposed to water. And just out of interest, the alveolar surface in a lung through its multi-folds and the amount of surface area produced by these alveoli is approximately equivalent to that of a tennis court. So that allows us to transfer oxygen and carbon dioxide in and out of the bloodstream very efficiently. Babies lacking this vital material would be born essentially healthy and then often succumb within hours or days because they had not yet secreted their own surfactant. This condition, which was originally known as hyaline membrane disease, was brought to prominence by the untimely death at 2 days of life of Patrick Bouvier Kennedy, who was the son of John and Jacqueline Kennedy. When it became apparent that NRDS was due to surfactant deficiency, the research community turned to seeking a replacement. The initial formulations consisted of the main lipid components of natural surfactant. So it was simply, they analyzed natural surfactant, and it turns out that about 3 or 4 different lipid components predominate, and Burroughs Wellcome at that time was one of the major companies to try to produce a fully synthetic surfactant based on that analysis. Mm-hmm. The original surfactants largely failed at the clinical level. Now, the product that was produced by Burroughs Wellcome was a product that was actually approved in Canada, and it did work. However, some of the very earliest attempts to do this did not work, and the results were so discouraging that the research in this area almost stopped until Dr. Gorin and Horning from the University of Buffalo began experimenting with a natural surfactant using an animal model. And I got to know Dr. Gorin and Horning. He was a wonderful man. He was a medical doctor who became an experimenter and a tinkerer, a producer of machines, and he was very inspiring for me. It turns out that trace level proteins, which had been overlooked in the initial formulations, were vital to the deposition of the surfactant monolayer. Clinical trials using surfactant formulations derived from natural source were successful with mortality reductions on the order of 30 to 50%. So it had a huge effect on the outcome of babies. Yes, it had a huge effect on the outcome of babies suffering from NRDS. And now ventilator techniques have become more optimized, and that sort of effect is not seen to the same degree, but it is generally considered that a baby suffering from RDS will benefit from surfactant replacement therapy. When I joined Fred Postmeyer's group in the early 90s, the hospitals were highly interested in obtaining surfactant as a research material, and a number of phase 2 clinical trials were underway, with a phase 3 being designed. These phase 2 trials were largely in the Toronto health system, based primarily at a women's college hospital. Let's see if I can find the rest of my notes here. The problem that the research group that I was part of encountered was that they simply did not know how to scale up the manufacturing of surfactant enough to meet the demand, which was already there, even though this product was not yet approved. The raw material is derived from bovine lungs at meat processing plants and required non-existent equipment for the extraction process. So my initial contribution was to design and build these extraction systems. With this issue dealt with, we were able to eventually form a company, which I currently operate together with a former researcher, Dr. David Bjarnason. The two of us built a production plant located in London, Ontario, which now employs about 50 staff. We are now in the process of handing over the day-to-day management of the company to a very capable cohort of younger staff. So that's the current state of our company, and it's been a long and very interesting road.
I guess I will have two follow-up questions. The first one, it's interesting to hear how you are creating your own equipment and how you seem to be kind of using things that you have created yourself. And I wonder if you think that has blinded you against, for example, things that are happening now, like the pandemic, that has allowed you to keep on growing without the influence from external factors.
I'm trying to understand your question. Do you mind just rephrasing?
Yes, I'm wondering if the fact that you are producing, like you develop your own equipment, you are not dependent on any external blockchain management that has allowed you to continue growing instead of doing the last years without kind of having to deal with importing things to Canada and so on.
Okay, yes, I understand now. Well, we obviously do have a significant number of items that have to be constantly, and consumables that have to be constantly used and purchased in our company. However, and in that, we tend to be very conservative in that we stockpile possibly a year ahead, typically on consumables. When it comes to the raw material, for example, the raw material extraction, the purification methods, etc., we are somewhat insulated because we have developed a high degree of resourcefulness. We actually are a company that takes our product all the way from a Canadian produced raw material at Canadian meat processing plants, and we take it right through to distributing it and selling it in multiple jurisdictions. The first jurisdiction, of course, was Canada for many years, but now we're in 21 other countries. But yes, that does allow us to be fairly independent when it comes to these sorts of disruptions of the supply chain.
So another question, because you already talked about the science and the surfactants and how it's used. I wonder if you, before the current pandemic, if you had tried to use that in other populations besides infants, or if the clinical trial that you are currently doing with older adults, it came just as a result of that.
Yeah. There were, after the initial success with natural surfactants for neonatal RDS, there was a fair bit of interest in using it in the adult population. And that met with a much lower degree of success. With the neonate, what you're dealing with is a healthy individual who was born simply too early. And so the very act of breathing becomes something which is harmful to the baby. And being ventilated, now the ventilators are much better than they used to be, but nonetheless, ventilating a surfactant deficient baby is hard on the baby's lungs. And so if you were able to increase the elasticity of the lungs of the premature infant, increase the compliance, allowing the ventilator pressures to be reduced greatly, and also the supplemental oxygen levels to be reduced greatly, it had a huge effect on the baby. Now, our interest in COVID work, and I'll just refer to my notes here, our interest came largely because of some anecdotal evidence that was coming out of Italy about a month and a half ago during their surge. And we had already known that there is some effect with adults if they are otherwise healthy. You're talking about an acute trauma to the lungs here with the COVID situation. So the trial that we are now supporting is looking into the potential efficacy of our product with COVID-19 patients who need ventilatory support. We received Health Canada approval a few weeks ago and are gearing up to treat our first patient. Using an approved product with a long history of safety in a fragile patient population made it relatively easy to obtain approval. And by the way, with respect to the neonatal population, we received approval from Health Canada in 2000. So we have been at market for about 20 years. And we've treated on the order of 160,000 premature infants in Canada and elsewhere. The coronavirus is thought to attack type 2 cells, which are responsible for surfactant production. With the loss of functional surfactant, the lung loses elasticity, making breathing more difficult. The situation is probably similar to that of the neonate, where a lack of elasticity or lung compliance requires that the baby, or in this case, the adult patient, be put on ventilatory support. A side effect of this is damage to the lung due to the inflationary pressure required to maintain adequate blood oxygenation levels. The question being asked by our trial is whether surfactant replacement can help to ameliorate this damage by increasing lung compliance. Anecdotal evidence, as I mentioned, coming out of Italy with about a dozen patients suggests that this might be a helpful intervention. The best results seem to be with patients who receive a dose of surfactant shortly after intubation, so very, very early on in ventilation. This trial is being conducted, the one that we're involved in, is being conducted out of several hospitals in London, Ontario. One obvious issue with such an intervention is that the per patient dose of this relatively expensive drug is much larger than that for a 750 gram premature infant. So the pivot, in our case, is one of increasing production capacity while reducing cost to meet this potential need. As opposed to a vaccine, our therapy would likely only be used for a fairly small subset of the most extremely affected COVID-19 cases.
Okay, so it's because of the production capacity that you need to keep it restrained to more emergency situations?
No, I wouldn't say that. I would say that it's because most people will not get to this level of need for intervention.
Okay, that's good to hear that it's not because of the production capacities. Yeah. So now that we are talking about COVID and the science behind the work that you both are doing, I'd like to move again to ask Dr. Ward about the scientific work that they're doing at Medicago. And I mean, we said it during the introduction and also at the description of the event that they're working in a plant-based vaccine. Perhaps you can tell us a little bit about that, and maybe if you can highlight some of the difference between that vaccine that is plant-based versus one that is animal-based. Is it more sustainable? Is it more easy to produce? I'll pass it to you to tell us a little bit about that.
Sure, thanks very much. The vaccine that Medicago is working on is a recombinant vaccine. So we are targeting the spike protein, which is the target for most of the vaccines that are being pursued now all over the world. The spike protein sits on the outside of the viral particle and is essential for the virus to bind to and enter into the host cell. Neutralizing antibodies against the spike protein can protect. So Medicago's vaccine is targeting the spike protein in a pre-fusion form. When the spike protein, which forms into trimers, they look like little clubs, when they interact with a cell, they undergo a really remarkable configuration change. And they actually form these spear-like structures that help the virus enter into the cell. The pre-fusion club form is what actually binds to the receptor. And so antibodies that target the receptor binding domain on the spike protein are thought to be likely to be the most effective antigens to elicit an antibody response that will protect. So what's unique about Medicago's system, the production platform, is that instead of using mammalian cells or yeast or bacteria to produce the protein, we actually use an Australian relative of the tobacco plant. It's called Nicotiana benthamiana. And we use it because it's a weed. It grows very well. It's an extremely robust plant. It's easy to transfect. So we transfect it with a bacterium that is defanged. So it's normally this bacterium called agrobacterium is a pathogen of plants, but we've tamed it down by removing some of its virulence factors. We insert the gene of interest into the bacterium, and then we essentially infect the plant. And we do so by inverting the plants into a bath containing billions and billions of these bacteria that contain the genetic information to drive the production of the spike protein. It's called agroinfiltration, and the inverted plant, you simply put the root structure under a vacuum, and then the leaves of the plant shrivel up like a sponge. When you release the vacuum, the plant re-expands and sucks the bacterial solution into its vascular system, so that virtually every cell in the plant's leaves is infected by the bacterium. We've then put the plant back into the sun, and it grows for 6, 7 days, and the virus-like particles form at the plant membrane, between the plant cell membrane and the cell wall. We digest away the cell wall, and those little virus-like particles float free, and that's our vaccine. So we're simply using the plant as the bioreactor to produce the vaccine, as opposed to using stainless steel tanks to grow Chinese hamster ovary cells or baculovirus infected insect cells. So all of these recombinant platforms are similar in some respects. Our upstream process is in a plant, and so is actually remarkably less expensive in some regards than the huge stainless steel bioreactor. Mm-hmm. It's also a little bit safer, frankly, because there are no pathogens that humans share with plants. And so the risk of an adventitious contaminating bacterium or virus in the plant material that could also infect a human being is largely eliminated by the use of plants. Mm-hmm. So it's a lovely and very flexible platform. In our greenhouses in Quebec City, we should be able to, if all goes well, be able to produce up to 2 million doses of the vaccine per month. And in our production facility in North Carolina, which is about 5 times larger, that number might go up to about 10 million per month. So we actually do have substantial production capabilities. In the global context, where we're not talking about making billions of doses, but certainly in the Canadian context, and even in the American context, the production of 100 million doses in the US facility would potentially be a substantial contribution to controlling COVID on both sides of the border.
That's really good to hear. I wonder, are there any others working on plant-based vaccines, or that's something very unique of Medicago?
Well, we're certainly the most advanced.
Okay.
Of course, I'm biased, but there's a company in the US called Kentucky Bioprocessing, which is using a very similar technology. And there are a few other places in the world that have the plant-based system going. Relatively few of these other places have the production capacity that we have, or that Kentucky Bioprocessing does in the United States.
Okay. And just another question, because I know that you were also, Medicago was also working on previous pandemic in the H1N1 and also part of the Ebola pandemic. I'm curious to know, is this a platform that allows you to easily put, depending on what type of pandemic it's going on, just produce other type of vaccines? I guess that's one part of the question. And the second one will be, what have you learned in the past from those previous pandemics?
Well, I think it's important to point out that pandemic vaccines and pandemic therapeutics are not a good business plan for any company, right? You need to actually have something that people want to use in the inter-pandemic period. Because Medicago's platform is a recombinant platform, in fact, we can make a wide range of vaccines. Virtually anything that you can make in any of the other recombinant platforms we can make. The plants appear to be particularly good at making vaccines and antibodies. And so during the West African Ebola crisis, we were asked to make the ZMapp antibodies for Ebola. One of the problems with antibody therapies, just like Dr. Nigh was saying, when you move into a larger person, you start to need really large quantities of material. And the Ebola antibody cocktails, we were talking about delivering 14, 15 milligrams of protein, while a vaccine is measured in micrograms, 15 to 20 micrograms of product. And that's where the production costs of the plant system, the advantages there become quite real, where you need to produce milligram quantities of an antibody. So yes, Medicago's platform can produce many different vaccines. At the current moment, our lead product are products against influenza. A seasonal vaccine is before the Canadian regulators now, a quadrivalent vaccine. We are what's considered pandemic ready for influenza, should the need arise. And we also are producing candidate vaccines for rotavirus, norovirus, and have several other vaccines in the pipeline.
Okay, thanks for sharing that. I see there's some other questions, but I will leave those from the audience. But I will leave those for later on. I guess just one last question: is there something that you think that either the government or Medicago or scientists themselves didn't learn from those previous experiences as working with Ebola or H1N1, that if you could go back, you said we should have done things differently based on what we learned at those times?
Well, so I think probably Dr. Nigh will agree with me on this point. We are vastly further ahead in 2020 than we were with the Ebola outbreak. And it was the Ebola outbreak that actually broke the myth that industry was going to solve the pandemic problems. Because with the West African outbreak, the major industrial partners basically said, we're not doing this anymore. We're not going to chase after each new pandemic, because what generally happens is the pandemic runs its course, and then nobody is interested in the vaccine or the therapeutic that we have spent hundreds of millions of dollars developing. And that was when CEPI arrived, the Coalition for Emerging Pathogen Innovation. And while CEPI has initially focused primarily on the production of vaccines for emerging pathogens, recently the European Union has taken up the idea that some of these therapies, like Dr. Nigh's for respiratory infections, might actually see broad application across many different pathogens that, in his case, affect the respiratory system. And so there is actually now a real private-public partnership that has developed first around CEPI and now through the IMI process in Europe around therapeutics, that we should in fact be preparing ahead of time and looking at platforms that might see application across multiple pathogens. That is a huge advance in the last 5 years. Everything that Dr. Nigh is doing now with BLES could easily be applied to other highly pathogenic respiratory infections like Nipah, like Hendra, like some of these others. And so I think there will be inter-pandemic support for the kind of work that his company is doing, and hopefully for the work that Medicago is doing.
Yeah, I can see that you have an entrepreneur on you, like really thinking outside the box and what are all the different possibilities that the work that each scientist is working on, not to focus only to a single solution, but really to think in broad terms. And I think these interactions and the possibility to do that really comes out of collaborating with different people, talking with different scientists and industries.
And support.
And support.
The support needs to be public-private partnerships for these pandemic efforts. And if Dr. Nigh's efforts succeed, I think it would be entirely logical to think about applying some of those same approaches, the same platform technologies, to other highly pathogenic respiratory illnesses. Obviously, BLES is moving from one neonatal condition to now a respiratory pathogen, but there are many respiratory pathogens. Yes,
and I would add that surfactant has long been considered a great way to, as a delivery modality for
absolutely
directly into the lung where it matters.
Absolutely.
Just simply because surfactant is really great at spreading throughout the lung surface. It's unmatched in that respect.
Yeah, drug delivery.
Yeah.
I see there's actually, before I move to the questions of the public, I wanted to ask one last question to Dr. Nigh, and it's perhaps if you can talk briefly about the roadblocks that you faced that you and your team faced when bringing BLES to life, and if you have perhaps any advice about for younger generations or younger scientists that would like to do these kind of things now, what do you wish that we would have established either from government or from accelerators or incubators such as District 3?
Yeah, that's a good question, and I think Dr. Ward has already touched on it somewhat. The biggest roadblock is moving from a university research mentality to a pharmaceutical compliant mentality. That was our big challenge that took us the better part of a decade, with a great deal of help from Health Canada in teaching us the difference between being a research group and being a pharmaceutical company. We were well placed in that Canadian hospital institutions were willing to participate in clinical trials and took an active part in procuring funding. There was a question about how did we manage to survive our pre-market period, and a large part of that has to do with the fact that at that time there was so much excitement and interest around the idea of surfactant replacement, and the early phase 1 and 2 trials were showing tremendous promise, and institutions like the Toronto University Health System, Royal Alexandra in Edmonton, BC Children's, many other hospitals were stepping up and simply doing their own curiosity driven trials. And so we had a real leg up there. We didn't have to do this all on our own. And these were influential people in the medical community who were applying for funding on their own. So we were also aware that the phase 1 and phase 2 trials indicated that our product was likely to fare well at the phase 3 level. So that was not as much of an issue as it might have been, because it at the low pre-statistical level, it was showing really great promise. So we really thought that our phase 3 was going to succeed. So for us, the biggest challenge was converting our mindset from that of a curiosity-driven research group to a compliant pharmaceutical company. The pre-market development stage took us about 7 to 10 years to complete. The trial period lasted about 6 years, and submission preparation with Health Canada review took another 3. Needless to say, this was a stressful period with major ups and downs, and the euphoria accompanying our notice of compliance was significant. However, when we have been through the process once, it is easier to do it again. At present, we're adding about 5 countries per year to our list of registrations. And as I was saying, we're in about 21 countries right now. With respect to the crisis, I would say that Health Canada has been very quick in answering the questions that we have submitted, and that our clinical trial protocol was approved in record time. I see no reason to think that this will continue, unfortunately, since after this crisis has passed. Long timelines for submission responses for anything from Health Canada has always been a problem. And this is not a criticism, it's simply the fact It's simply the situation that we exist with in Canada. So patience is absolutely essential.
I see.
Just two words. Dr. Nigh moved past the word compliance just with the word. And academics have no comprehension of what it takes to be pharmaceutical compliant. From the first biochemical experiment you do all the way through to the last patient you enroll, the level of meticulousness and compulsivity that is required to be compliant is beyond the comprehension of most academics.
Yeah,
it is a different world.
Yeah, and that was the world that it took us 1 decade to move from one to the other.
That's not unusual.
Yeah. And so
yes.
I would add that any sort of incubator project should really include mentorship with people who have made that move, because without that, there are so many pitfalls.
Absolutely.
Yeah.
Thanks for pointing that out. We're actually working, since we're opening the BioHope, in recruiting new mentors. So we can take that conversation offline after, if you're willing to volunteer or spend some time with some of our startups. But that's definitely something we're paying a lot of attention to, because especially when you're dealing with companies that are so specialized, you do need somebody that is specialized as well, and then that knows this other business world. So it's not only about the science, it's about finding somebody that knows about the science, the business, and has passed through these, through all of these challenges as well. And I saw that Dr. Ward was kind of nodding every time Dr. Nigh was commenting about the roadblocks and how difficult it is. Maybe just, would you like to add something on that? Or
no, I don't think I need to add anything. Simply to say that it is, I don't think that any idea has gone from the idea to a product that's used in animals or people without encountering significant problems. Problems in production, problem in contamination, problem in just about anything you can think of, all the way up to regulatory and then even distribution. You need to be at the level of the small company, you need to be a jack of all trades and enormously flexible. And then as the company gets larger, Medicago, during the time that I've been interacting with them, has gone from 17 people to just under 500. And it's actually quite difficult as you move from the 17-person phase to the 500-person phase to delegate and to give up some of the things that you had full control over in the little tiny company. But you have to, because you can't be looking at everything at all times. And that involves training, mentoring, letting go. Some things that academics are not very good at. Honestly, the history of academics surviving in industry is not good. It is the usual pattern for the academics who start a company at some point to find themselves leaving the company. So Dr. Nigh and his colleagues are unusual in actually moving, even if it took 10 painful years, moving from the academic environment to actually running a company. That is not an easy transition for academics to make.
I fully agree. It's a big challenge, and there are many pitfalls, but if you can make it, it is an incredibly rewarding career.
I guess we're about 5 minutes to the end, but I think yourself brought the discussion to the final topic we wanted to cover, and it is perhaps ending on a more positive note. What kind of advice can we give to young scientists that are interested in working in entrepreneurship? And what can they do to be better prepared? Who who could they contact with? Anything that comes into your mind?
Go ahead, work.
You need to be open. I think that there are increasingly opportunities for graduate students and postdocs to spend some time in management schools and learning sort of some of the corporate vocabulary. I think that the MyTax programs actively encourage people to look for opportunities that bridge academic and industry projects. Those are very good ways to start. And frankly, they are easier to get in many circumstances than a classic academic PhD or postdoc, precisely because of the larger resources that are available within industry. What I think is really important for PhD students and postdocs is to recognize that doing that kind of a PhD or a postdoc, it's not a one-way street. It doesn't mean that you are out of the academic stream. If you choose your projects well, you actually can learn an amazing amount through one of, for example, the MyTax programs that you can then bring back to a university post, and you will be welcomed, actually. If you are applying to a junior faculty position, and you bring industry experience, you will stand out from your peers. You can publish and publish well working with industry. You need to make those arrangements up front, but you can have every bit as good a CV working in an industry academic collaboration as you would have in a pure academic environment. I'll leave the last word to Dr. Nigh.
Yes, that's, I couldn't agree more. And you also made the point earlier about being willing to do whatever the task is that has to happen. And that is so critical. I mean, you cannot, if a job is there, then, and there's nobody else to do it, no matter what your credentials are, you have to do it. And if you're willing to do that, your odds of success are going to be a whole lot greater. So I would have a couple of points that I would suggest. And one of them is around this issue of patience. It took us 10 years to get from a university lab to a Health Canada licensed product. And during those 10 years, there were many times when we might have wanted to just quit and do something else that would have had quicker rewards, but we didn't. So our experience, which perhaps isn't the experience of everybody, but our experience says that if you are patient, it will pay off in the end. Another thing is, don't get too mesmerized by the early seeming success of something that's happening at the bench. Our experience was that something that had a very high degree of effectiveness at the phase 1 and 2 clinical level still required a lengthy phase 3 trial to show statistical significance. It took a long time, even though at the earlier level, it looked like this was a complete winner, and it was. It's just that to show that statistical significance that Health Canada requires, it still is it still takes a lot of time. Think very carefully about the level and type of financing you need. Yeah. The idea that you have is precious, and there will be plenty of people who will want a piece of it. Remember, you only give away your shares once. Finally, don't lose your integrity. We are here to provide a service first, and secondly, to make money. This attitude is not only good for you personally, it is, in my experience, a good business strategy. Marketing has a much shorter effectiveness timeline than reputation.
Thank you so much for this lovely ending note. And so thanks again really to the speakers and our participants for joining. I really hope that you found this dialogue practical and that you like the advice. I shared my email at the beginning. We will share it again in the chat if you have any other questions. Otherwise, thank you so much for joining.
Thank you, Anna.
Thank you, Anna.
Pleasure to meet you, Dr. Nigh.
Yeah, you as well. Okay.